Welcome to the Modern Biopharma Podcast. The world of today's medicines is big, complex, and constantly changing. Our goal is to give you insight into the people that are manufacturing the medicines people need every day, whether it is the people taking care of these facilities, or those building them, or those maintaining the quality and integrity of every dose that is delivered to the patient. We interview the people that are making it happen and give you a chance to get to know them while we're at it. Thanks for saying hi. Hey everyone, welcome to another episode of the Modern Biopharma Podcast. We are happy to have on today Brian Hagopian with Clearwater Consulting. Brian's an industry expert up here. I know him up in New England. He's known much further beyond New England, but he's got 30 years of high purity water system design, construction, commissioning, and validation experience. Delivered a wide range of solutions to premier pharmaceutical, biotech, semi-conductor research, and other biotech clients. He's a recognized industry leader and active volunteer with numerous professional societies. He was the past president of the Boston chapter of the ISPE group up here in New England. And he's a contributing author and lecturer on topics associated with USP purified water, water for injection, and semiconductor grade water. How you doing today, Brian?
Doing great. Happy to be here.
Awesome. Glad to have you. I hope that wasn't too long of an intro, but you got a lot of background. So we're excited to have Brian on today. What we're going to do is we're going to be doing a 3-part series with Brian just to talk about some things in regards to water in the pharma space and maybe even water on the edges and how they might relate to the pharma space. And Brian has a lot of experience in many different kinds of accounts, and all that comes together to make him extremely well qualified to talk about what can be done, what should be done, what might be done in the pharma water space. So that's what we're going to do. And today we're going to be talking about the microelectronics water and, what pharma could learn from them, what maybe pharma has learned from them. And Brian's got a bunch of experience there that we're going to talk to today. So first off, Brian, do you know when we met for the first time?
Did I know you were going to put me on the spot? When you tell me the story, I'm probably going to remember it. But
no, it's not bad or anything. No, actually. So Brian he does a ton of stuff with ISPE. That's actually where I've known. It's where sort of our paths have crossed most often. And he taught a CPEC class, a Certified Pharmaceutical Industry Professional, if I remember the acronym. He was teaching that, and I signed up and did that class, and he was teaching that in I don't know if it was the basement, first floor of Biogen in Cambridge, back in 2013, I think.
Yeah, 2012, 2013.
Yeah, I think that's it. So that's where I first met you, and you were teaching that class. I learned a lot. I didn't actually finish it. I was I don't know what happened. I didn't finish it. It was really way above my head when I first started. May still be. But I remember one of our classes, we were sitting in there and all of a sudden we heard what sounded like a train went underneath our class. Like it felt like there's a train, and everybody's phone started to ring in the middle of the class, and it was an earthquake in Boston. Do you remember that?
Yeah.
It was the weirdest thing. Anyways,
It was.
I'd never experienced, I'm not from California, so I never experienced that. And I remember like, what? I didn't know there was a subway underneath Biogen. What do you know? So anyways, little background. We won't do that on the rest of the episodes, but that's the background for Brian. And
so let me just clarify though.
Okay.
I didn't teach the study group. I was a facilitator.
Oh,
the material was pre-recorded. ISPE International provided it. And I was there just to make sure everything flowed and we had discussions and what have you. So I can't take credit for teaching it, but I will take credit for being a facilitator.
All right. Well, you were a teacher as far as I was concerned, because I knew so little. Anyways, yeah. And Brian does a ton of stuff. He's probably an ISP in the most recent years, I think, with college kids and trying to get a lot of the local universities involved in ISP. Maybe I'm wrong, but I think that's where you've spent the majority of your ISPE time. Is that right?
Yeah, I mean, of late, yes. I spent a lot of time early on with the product show and helped to move it from Cambridge to Newton, and then from Newton to Gillette Stadium to help it kind of grow into where it is. I've been involved in the educational program committee. And when I was president of the chapter, I realized that we weren't doing such a good job getting students involved in our industry. And I thought I had the bandwidth and the knowledge and the network to be able to do something about it. So I kind of made it my mission for about the last 6 or 7 years. And I've just stepped down from that role. I'm still doing one of the major events, but I've turned the reins of the committee over to the capable hands of George West in the past year, and he's been taking it, and running with it from there.
Yeah, that's great. And I think, we'll see what the show looks like this year, but we've actually engaged a group associated with high schoolers in Massachusetts to try to get them involved too. So it continues to grow, just like the product show has grown since you did it. Now, hopefully the outreach to students can grow as well. So thank you for all those years of serving the community here in the Boston area. All right, so what we're going to do is I got a bunch of questions for Brian today. We're going to talk through in relation to pharma versus microelectronics water. This came about because he just recently did a webinar for the ISPE group up here in Boston, which I attended. It was quite good, and he covered a whole bunch of stuff. But as is always the case in these, there's always a whole bunch that he couldn't get to, and so I thought, hey, let's talk to Brian and pick his brain a little bit further about some of these things. So what I wanted to go to first is let's just talk about the industry, the microelectronics industry, and in relation to some of the drivers associated with how we come about determining water quality. So we have a USP that defines water qualities for purified water and for water for injection, in our world we're a little bit more familiar with. But they got the same kind of thing going on in the microelectronics world. So can you talk about what are the main drivers first in the pharma market, and then maybe we'll work from there over into the microelectronics market?
Sure. So in the pharma market, the main concern with any water-based product is that it not have anything in it that is going to make people sick. If it's an injectable drug, there are different requirements for things, for example, that we would drink, that we would ingest, as opposed to things that get shot in our arm. And that's kind of the major differences between where we need to use USP purified water versus USP water for injection grade water. And the major concern, the major concern is bacteria. There are other things, there's a conductivity requirement, which basically means that you've got to get most of the minerals out of the water. There's a organics requirement, which is described as TOC, and I'll talk a lot more about that a little bit later. And for injectable grade water, there's a requirement for endotoxins, and endotoxins are kind of defined as things that, if injected into your system, would cause you to get a temperature. Okay, bacteria particularly, there are some bacteria that can cause problems, and obviously we want to eliminate those. So, in the pharma industry, the major concern is getting most of the minerals out, keeping the organics relatively low, and getting the bacteria out so that the products can be safely administered. Now, the microelectronics industry is, it's on a different level. These are the folks that make integrated circuits, the stuff that makes all of our computers run, our smartphones run, and what have you. And, these are the semi-conductor chips, and the industry has evolved in a way where they're literally writing little tiny lines, and they're separated by less than a micron in distance. And any type of contaminant, doesn't matter if it's a particle, doesn't matter if it's a bacteria, it can be anything. But if there's a particle or a bacteria that bridges across and kind of creates a short circuit between two of those lines, the whole chip doesn't work. Okay, so the microelectronics industry is very, very concerned about bacteria, dead bacteria, fragments of bacteria in addition to any particles that are floating around. And because what they make is a silicon-based semiconductor, and silicone is a unique material in that sometimes it's conductive relative to other things, and sometimes it's not conductive relative to other things. So that's why they call it a semiconductor. Some of the times it does, some of the times it doesn't. But the majority of materials that are kind of implanted or built in, and I might be misstating things a little bit here, contain metals. So it's extremely important in the microelectronics industry that there are no metals in the water to interfere with, all the things that they're trying to do as they build these integrated circuits. So if you go into a microelectronics plant, you'll see almost no stainless steel. Whereas if you go into a pharmaceutical plant, you're going to see almost exclusively stainless steel. If a microelectronics plant tried to convey their water through a stainless steel piping system, all of their products would fail. All the products would fail. So, it's a different industry, and they have much different drivers. They don't care whether there's a living or a dead organism or a particle. Any of those things can cause defects, and they're all about defect reduction. Okay.
So can you just talk to that real quick? So again, in the pharma market, when we're measuring different types of things, we're looking for bacteria, right? We're actually sampling and looking for colony forming units, and that's important. We're looking separately for endotoxins. There's a different test for doing that. Like if those mattered in the semiconductor world, did they actually look for that, or what are they looking for in comparison?
Well, so in the, if you look at what all the pharmacopoeias tend to require, and how do I want to say this? There are across the world over 140 different pharmacopoeias. The big three are the United States, Europe, and Japan. And now people are obviously recognizing China, Brazil, India. Okay, but there are literally 140 pharmacopoeias. Most of them do not have a requirement for bacteria, which I find really interesting. They've got requirements, and there's usually a little bit of difference between what's required in Europe versus what's required in the United States versus what's required in Japan. And there's an effort underway to try to harmonize all these things, but there are some differences. So, locally, those are things you have to be concerned about. If you're going to be selling a product in country X, you have to be concerned about making sure that if you're going to make pharmaceutical grade water there, that you follow all of the requirements of their pharmacopoeias. Okay. So, so I'm sorry, I just lost track of where I was going with that.
Well, we're talking about, my question was more in regards to this was tracking in the right direction. You just went. So, why does the semiconductor industry, are they measuring for bacteria and endotoxin? Because that's important to them, but are they measuring for that like the pharma does, or do they measure different things or a different way to ensure that that's not present?
Measure the things that they're concerned about. They're concerned about particles. Okay, and they're concerned about particles in a specific size range. They're also concerned with a whole slew of different metals because, conductive metals, if deposited in a place where they don't want them, it's going to ruin a chip. Okay. Now, interestingly, a couple of years ago, I was invited to make a presentation at an ultra pure water conference in Portland, and this was 2 or 3 years ago. And they had a pharmaceutical track and they had a microelectronics track, and there were 2 different groups of people. And during breaks and intermissions, you had a chance to mingle. And because I play in both industries, but I was there for the pharma, I wanted to kind of get a feel for what they were talking about in the microelectronics industry. And, the pharma talks were all about bacteria and control and different ways of doing that. In the microelectronics industry, they've been able to generate water that is pure enough that literally they don't worry about bacteria. It's not a concern. They basically have all said, it's not a concern. Now,
Huh.
wouldn't it be great if the pharmaceutical industry could get to the point where they could say, well, yeah, bacteria, the water is clean enough, bacteria is not of concern.
Yeah, we care about bacteria, but it's never going to exist in this based on how we make our water. Yeah. Wow.
Well, and again, the pharma industry kind of has taken a different perspective. The kind of going back, if we turn on the Wayback Machine, the tried and true way and the method everybody recognizes to produce injectable grade water is by distillation. You heat the water, you boil it off, you keep it hot, and in theory, you can't grow any bacteria. Okay, and that's worked. And so you get a lot of why would we stray from that if it works? And people haven't really put in the I don't want to say diligence, that might not be the right word, but people haven't really kind of boiled it down to, why do these things happen and what do we need to do to control them? So, as a chemist, I've come to realize, and I play with a lot of microbiologists as well, that things live in equilibrium. We live in equilibrium with our environment. Bacteria live in equilibrium with their environment. And the easiest way that I could kind of explain something to you is, let's say, for example, I were to take you and put you in a helicopter and drop you on top of a mountain and say, I'm gonna come back in a week. Okay, you have the clothes on your back. I didn't give you any food. You're on your own.
It's
So
no
what's the
good,
first thing
Brian.
you're gonna
I'm not a good camper. This is not a good analogy for me, but I'm going to let you go with it.
do? Well, so what's the first thing you're going to do? Okay, you're going to look for food and you're going to look for shelter, right? Because you got to make it through a week. Well, if we boil it down to the cellular level, that's exactly what bacteria do. When we have a brand new bacteria that gets introduced to a water distribution system, it's looking for food and shelter. So the first thing it does is it finds a place where there isn't a lot of flow, where it can kind of settle down and it'll stick to the pipes, and it's capable of building its own shelter, and that shelter is called biofilm. So it kind of builds its own little protective cocoon or barrier around itself, and then it goes looking for food. And when I say it goes looking for food, it looks for food in the water that's flowing by it. And we've got lots of water flowing through these pipes. And if there's enough food, and I look at TOC as a measurement of the amount of organic matter, because that's what it is, to measure the food supply that's available to bacteria.
Mm-hmm.
If there's enough food, guess what? Bacteria is going to be able to easily absorb nutrients. Bacteria is going to grow. Bacteria is going to multiply, and bacteria is going to they're going to kind of send out scouts and go and populate the rest of the system. So in the microelectronics industry, they've recognized that if they really restrict the food supply, okay, what's going to happen? What's going to happen is the metabolism of a bacteria is going to slow down and stop. Now, you ever been on a diet? Tried to lose some weight?
Oh, this is not
Okay,
good. 2 months. 2 months at home. Oh, man.
I retract the question, but have you ever tried, let me ask it a different way. Have you ever tried a starvation diet?
Oh, yeah. Yeah.
Where you say, like, okay, I'm not gonna eat anything for a little while, and you really cut down. Well, something funny happens with your body. Your body says, hey, you're not giving me any food, so completely out of your control, your metabolism slows down, right?
Yeah.
Okay, now that's exactly the way bacteria behave. We regulate their food supply, we cut off their food supply, we reduce their TOC, and now all of a sudden, they're not going to multiply, they're not going to divide. In fact, they're not going to grow as fast because there's no nutrients there.
Right.
In the microelectronics industry, what they've done is they've literally pushed this equilibrium to the extreme, and they've taken the food supply away. They maintain water that's got TOC levels less than 1 part per billion.
Wow.
And bacteria can be controlled. And in fact, I've actually built systems like this. So I've got a system that I've installed, and I still do a little bit of work with the client. But I installed the system in 2002. That's 18 years ago. It was sanitized when the building was first commissioned, and it's never been sanitized since. And they don't have bacteria.
Hmm.
Now,
That's a
a lot of
long
people,
time.
yeah, and it's an ambient temperature system.
Right, which is everybody's fear, right? In pharma
Right,
is
right.
how can we do this in ambient and never have bacteria? Everybody's kind of scared to death of that right now. So
Yeah. So it can be done, and in certain in certain occasions, I've had the opportunity to do it in a pharma application, and it works. It works.
right.
So
So
um,
so just since we're talking about this and going down that route of, let's get, TOC, I mean, everything else, but TOC from the bacteria perspective, which it's pretty easy to get conductivity down to an acceptable level for a pharma system, like, right. Conductivities for semiconductor systems are a little bit different. But how do you get those TOCs down to that level? Like, what are the process steps to be able to achieve that 1 ppb? I mean, that's basically you're at the undetectable levels at that point, right?
I mean, you can certainly detect them. There are TOC analyzers that have been developed, and in fact, the microelectronics industry has pushed a lot of the instrumentation that's been developed to to the point where it is. How do we get there? There are a couple of different ways to get there. One of the main I don't know, I don't want to call it fallacies, but one of the things that they do in the pharma industry is once something goes through final treatment, and that's usually like a distillation or a 2-pass RO or something like that to make WFI, and it goes into storage, nothing is done to that water other than heat being applied.
No further process, right? Or
No further processing. And in fact, there are times where an inspector would look for additional processing as a sign that there's a deficiency in the system somewhere. Okay. People in the microelectronics industry don't look at it that way. In fact, they do polishing, what they call polishing, trace removal of materials all the way through their distribution systems.
Yeah.
Okay. And so there's a storage tank, and there's a bunch of other treatment steps that happen before this water is distributed to the building. So, that's one thing that kind of would challenge some of the standard ways of doing things. And, pharmaceutical people are, and it's human nature, creatures of habit. We've done this, it works, right? We use stainless steel, and if we have a problem, we blast it with heat. And it's worked this way, for the last 80 years, and it's going to work again for the next 80 years. What they don't realize is that if their TOCs are in the hundreds, there's enough food there that we're going, if bacteria are introduced, it's going to become a problem.
Yeah.
And so what's the solution? Is the solution, should we sanitize with heat more frequently? Or maybe get to the root cause of why we're having so many bacteria, because there's so much food. Can we cut down on the food? That's a spot where, for example, a process like ozone would have some value. Ozone is really, really good at reducing TOC. And, that's a process that has had limited acceptance in the pharmaceutical industry. People don't like to use demineralizers because of the fear that they grow bacteria. And in certain instances, that's the case. But in other instances, it can be used very effectively with something like an ozone generator or a 185 nanometer UV unit to effectively eliminate TOC and to control TOC.
So I hear it said, I'm pretty sure I probably said it at some point, is that in pharma systems, there's pretty much always guaranteed some amount of like film or, slime layer in the piping, especially in like an ambient system. In these semiconductor systems, that doesn't exist.
I can't say that it doesn't exist. I don't think it does.
You can't get in there to check it out because they haven't opened it in 20 years.
Well, You know, I've worked with a few semiconductor plants, and when they bring on, you know, a new section of piping or a new fab or whatever, they go through a sanitization process, and then literally they go through a flushing process
Mm-hmm.
where they may flush for 2 weeks, 3 weeks. Okay. And the other thing with microelectronics water is they reclaim a lot of their water. So they'll take a lot of their uncontaminated water and return it right back to their system and reuse it again. So, you know, there are a lot of different things going on that wouldn't necessarily be done in pharma. But then again, in microelectronics, we're talking, you know, literally hundreds, if not thousands of gallons a minute of water being produced and distributed.
Yeah,
So
much on the big semiconductor plants, those are doing millions of gallons a day sometimes, right?
yeah,
Versus pharmas rarely are doing that.
right.
Yeah. That's interesting. Yeah, something like reusing the water, you know, we're not allowed to really in pharma, right?
Correct.
Because of the rules set, because once you've done all the process, it's no longer you know, fits the description of the water it has to use when you make it in the first place.
Well, you know, one of the biggest challenges that we have in pharma is that it's a regulated industry.
Mm-hmm.
And it's making products that are used, that are injected into humans. And there are, you know, there are much stricter requirements for documentation. And once you validate a process, you are encouraged not to make any changes to that process, or a revalidation is required. In the microelectronics industry, if they discover that there's a better way to do something and you can improve product yield, they're going to go right ahead and do it.
Right. So I just want to hit back on the thing I asked, because you covered it sort of like with a broad brush. But when you talk about, like, say, a distribution system in a semiconductor plant, how do they maintain this really, really low levels? There's a few different components they use to ensure that, right? And again, you're saying in pharma, like, if there's anything, that means there might be a problem on the front end, so we try to avoid it. But what are those things that they're typically using in a semiconductor system to make sure particles are low and TOC stays low and all those things?
Oh, it's not uncommon for them to use point of use ultrafilters or point of use reverse osmosis units. Mm-hmm. to keep the particulate materials out. You know, all of their piping systems are done in fluoropolymers, either, you know, a Teflon derivative or a PVDF material. These are things that are extremely pure, that do not extract. You know, and all these systems are run at ambient temperature. Okay, stainless steel just wouldn't cut it in the pharma industry, because, you know, they're running 18 megohm water, and it will, you know, that purity of water will extract, you know, parts per billion and parts per trillion levels of these metals, and that will adversely impact the quality of a semiconductor product that somebody's trying to make. Okay. So they do everything in their power to get rid of stainless steel wherever possible.
Yeah, I don't know if I've ever even seen, I don't know if I've ever seen a point of use RO on those. I guess that makes sense. I've seen the UF systems. They have them on the loops too, right? Like,
yeah. Oh, absolutely.
I remember using a 15 nanometer particle filter on the loops, and that cleaned up a loop really well. Took the particles down a long ways. So, yeah, and they got resin. They got resin on these loops too, right?
Yeah. Oh, absolutely.
Yeah, UVs. Although there's probably stainless steel in the UVs, I suppose.
There are. Yeah, I'm not sure where they're at in that regard, but, you know, they've figured out a way to manage, you know, keep TOCs extremely low, and, you know, the bacteria goes away. I mean, the whole idea is, you know, frankly, bacteria have to be able to form a biofilm, have enough food to grow, build enough of a population where they say, okay, now it's time, we can send out scouts for the next, you know, to populate other parts of the system. If you have a bacteria in a system that's starved for TOC, maybe it adheres to the side of the pipe, but there's no food, so it's really not going to grow. Or if it does, it's going to grow really, really slowly, and it's never going to get to the point where it's going to release bacteria.
Mm-hmm.
So if it's stuck in biofilm, and I think you probably know this, and most people, if they don't, they should, that for every bacteria that you find in when you culture and test for bacteria, that probably means there's at least by a factor of 1,000 to 10,000 that many more bacteria present in your water system in the biofilm.
Wow.
Okay, so yeah, and it's kind of so when when we're testing a purified water system and the bacteria counts get to be 50 CFU per mil, we're going to be sanitizing because there's a lot of biofilm in that system and it's releasing a lot of bacteria.
Right. Wow. Yeah.
The whole idea is low TOC. You literally starve the bacteria, and they're not going to grow. They're not going to multiply. They're not going to release bacteria. And if there's any present, they're going to be stuck, somewhere to the sides of the pipes and in the biofilm, and that's it. You're not going to see them.
Got a failure to thrive right there is what you have, right?
Yeah.
So we gotta get close to ending up on this one, but I wanted to ask a couple other questions real quick. So, semiconductor microelectronics, they're not really regulated the same. I'm just curious, what kind of documentation requirements do they have for those kind of systems? So they're not regulated, so maybe they can just do whatever they want. Is that really true? Do they have the same kind of documentation, different? I would expect a lot of money's riding on these things. So,
well, there is a lot of money riding on, but it's not a we don't have lives at stake.
Yep.
It's product and it's profits and it's efficiencies. So they're all data driven. Make a change. Let's change the process one way and let's look at the data. Did we make more product? Do we have a higher yield? Whatever the metrics are, and that points them in the direction that they need to go to. So it's a much different, it's a much different thing. We don't have patience to worry about.
Yeah, and they can watch it real time. They can watch changes basically going on,
yes.
right? Yeah. So it is. There's not this thing that's going to happen down the line as much. So
right.
Interesting. Okay, and then just something I think. Since they seem to be driving a lot of this, we have had some changes in the water business for pharma recently, in regards to being able to use membranes more now. How do you foresee maybe some of what the microelectronics industry has been doing, being able to sort of educate the pharma market moving forward as we're sort of embracing membranes, especially for water for injection, in the future?
Yeah, I do think that every pharmacopoeia recognizes that distillation is in the United States, they consider it the gold standard, but they were smart enough to be able to say, listen, we recognize that other technologies may become available that are capable of producing this grade of water. And as long as you can validate that your system's under a state of control and that you can reliably and consistently produce this grade of water with these technologies, that's acceptable to us. Okay. Mm-hmm. Some pharmacopoeias are much more prescriptive. Some of them say you have to distill. Some of them say, if you don't distill, you have to do RO followed by UF. There are different, and again, this is part of, the harmonization challenge. But I think more and more, as we're going to see, people are recognizing that there's more than one way to make WFI water.
Yeah. All right. Cool. Do you see any other sort of risks in the pharma market associated with these changes? Maybe practical risks rather than just, yes, there's always risk because you change your process. But I'm curious, do you see practical risks for like the end user, like as they're putting in a different kind of system than they have before?
Well, I think just kind of by the nature of the business, pharma companies they tend to be much more risk averse because patient safety issues are paramount. They take precedence over everything else. And, if we've done it by method X, for the last 45 years or the last 85 years and everything has worked, why would we change from a tried and true method?
Mm-hmm. If it ain't broke, don't fix it.
Yeah, I mean,
yeah.
I do, however, think that some of the things that the microelectronics industry has uncovered as the mechanisms kind of going on behind the scenes can be of value to the pharma industry. Ozone is an example. It hasn't had a lot of widespread acceptance, but ozone is a technology that's used widely in the microelectronics industry, and it's very effective at reducing bacteria, eliminating bacteria, chewing up biofilm and things like that. It's had limited acceptance, but I think as time goes on, we're going to see increased acceptance of it, as we're going to see increased acceptance of alternative technologies for producing WFI water.
Yeah.
Based on data and performance.
Yeah. Yeah. That's I think I remember you telling me, it was a few years ago. I remember we had a conversation about ozone and you said, it's been the next big thing for the last 30 years. I think that's how you described ozone. And I think it actually finally is. It's in the pharma world anyway. It seems to finally be popping.
It takes a long time for any of these changes to take root and to take hold. And I've watched ozone evolve as a technology and watched the impression of ozone. I mean, what it does hasn't changed, but what its impression is in the pharma industry, and it's slowly but surely becoming a little bit more mainstream, a little bit more acceptable. So
yeah. Well, I know I've seen it, semiconductor, they have it in the beverage world. They, food and beverage, they use it a lot, right? It's in all the bottled water plants that's in the water when they put it in the bottles most of the time. So
absolutely,
it's very common there. And I think in the pharma market, I can attest to the fact that I'm seeing a lot of systems getting designed with ozone right now. I know of multiple on my desk. So they keep popping, and I think it's good. I think it's, on its face, I always get excited about people being willing to look at a technology that they should have been looking at for a long time. So I'm glad that that's happening. And and I know, like I said, you've talked about it for a few years. I've heard you say things about it, saying, "Hey, this is a good one. We just need to understand it, embrace it, and use it where it makes sense." So.
Yeah, and I think part of the problem is that people may have jumped in a little too fast and a little too hard and didn't really understand all the issues with ozone. Because if you over-ozonate, the excess ozone is going to go and attack everything. So it's going to attack mechanical seals on pumps. It's going to attack elastomers. And, the next thing you know, you're going to have a bunch of leaks and you're going to violate the integrity of your system. And, there are all sorts of negative consequences of that. So, you have a couple of those experiences and ozone gets a really big black eye.
For sure. And going back to your previous comment about, hey, we've been doing this for 50 years this way. Why would I change? Well, all it takes is one of those experiences. And guess what? Nobody's changing again for a while, right?
We're going back. That's right. We're going back to the way
that was fun. Let's use that heat exchanger. So, all right. Awesome. Well, this was great. Thanks, Brian. We're gonna move on to some other issues here, but this was a great recap of sort of how the influence of microelectronics industry on water treatment and how the pharma space can learn from what they're doing. So, thank you so much for today.
All right, great. Happy to be here.
Thanks again for listening to the Modern Biopharma Podcast. Please reach out if there is someone you'd like us to interview or a topic you'd like us to discuss. We will make it happen. And don't forget to like us on iTunes or subscribe on YouTube. It helps us get the word out. See you next time.